Welcome to AP Biology. Today I want to talk to you about the flow of genetic information from a gene, which is a piece of DNA, to RNA to a protein. So the general overview is that DNA codes for RNA and RNA codes for a polypeptide or a protein, right? So that's not always the case. There are viruses like retroviruses that have RNA as their genetic material, which then codes for DNA, which then codes for RNA, which then codes for polypeptide. Sometimes the DNA actually doesn't end up coding for a polypeptide. Maybe it might code for a different type of RNA. So what I've drawn out here, the RNA is messenger RNA, but DNA also codes for transfer RNA, right? And that transfer RNA doesn't directly code for a polypeptide. So there are other types of RNA, but the general gist is that DNA codes for messenger RNA, which codes for a polypeptide, with a few other types of RNA in here that we can add in later. So the instructions for making you are written in your DNA code. One important point, though, is that when I look at you, I'm not actually looking at your DNA. I'm looking at what your DNA has coded for. So we just said that some DNA codes for messenger RNA, some for other RNAs, and then messenger RNA codes for polypeptides. So when I look at you, what I'm looking at to some degree is your polypeptides, right? Those are folded and you have a protein, but that's not all of you. So you are also made of other molecules too, right? So you're also made out of carbohydrates, and you're also made out of lipids, right? So the four macromolecules, the four big molecules are lipids, carbohydrates, nucleic acids, and proteins, right? Or polypeptides. And so how are all of these made? Well, we're going to spend this whole unit talking about how DNA makes RNA and RNA makes proteins or polypeptides. Um, so we'll talk about these two just very briefly here. So how is DNA made? Well, DNA is made in this process of replication where you have special enzymes like helicase and DNA polymerase and ligase and topoisomerase and primase that are needed to make more DNA. So each side of the DNA serves as a template for the other side, but you need enzymes to get you there. And then DNA to RNA also requires enzymes. So we're talking about RNA polymerase in this case. And then RNA to polypeptides, well, that happens on a ribosome. But even ribosomes have catalytic functions. They function like enzymes. So they are made out of protein and ribosomal RNA, and they catalyze the formation of a polypeptide from RNA. So how do you make carbohydrates and lipids now that I know how to make nucleic acids, right? DNA and RNA are both nucleic acids. And polypeptides, right? If you fold it the right way, that's a protein. Remember, just if you don't remember the difference between them, a polypeptide is a bunch of amino acids in a line. If you fold them just so, and you make some beta pleated sheets like this, and maybe some alpha helices like this, and then they fold over on themselves, then you might have your final protein made. Or maybe you need a couple of polypeptides folded together to make your protein. But the polypeptide, or folded just right, maybe within another chain, is a protein, right? And we're going to spend the whole unit talking about that. But how do you make carbs and lipids? Well, some proteins are enzymes, and you need, that's supposed to say enzyme. Let's see, let me maybe use a smaller one for that. Some proteins are enzymes. And of course, you need enzymes to make DNA and for DNA to make RNA, and you need catalytic molecules to also make proteins. But some enzymes will actually put together monosaccharides to make the right carbohydrate for you, or they'll put together a glycerol and fatty acid to make your lipids, or a particular lipid that you need to make. Or they'll put a lipid onto a protein, and then you'll have a lipoprotein, or they'll put a carbohydrate onto a protein and then you'll have a glycoprotein, or you can have a carb and a lipid for a glycolipid, right? So anyway, enzymes are really the important thing here. That's what makes all of you. And you're made out of all of these materials. We're going to spend this unit on DNA makes RNA makes proteins, but also know that the rest of you, the carbs and the lipids in you, are actually protein products as well, because your proteins can be enzymes and the enzymes can produce the lipids and the carbs that you need. Okay, so proteins are usually, not always, but usually the link between genotype and phenotype. So our last unit was on genetics, and genotype is your DNA, right? Do you have a big P or a little p, for example, for flower color? And phenotype are the physical characteristics, which might be what you look like, or it might be something microscopic. But in this case, it's purple flowers as opposed to white flowers. So let's say this piece of DNA codes for the purple color. That's your genotype. That allele would be a big P, and maybe you've got two of these chromosomes, and let's say you've got two big Ps. Well, they are going to code for a protein that helps to make a pigment, and then that pigment makes you purple. So your genotype is the DNA, and your phenotype is your physical characteristic. So the central dogma of biology is that, where's my pen? Is that DNA makes RNA, which makes proteins. That is true, but we should probably call them polypeptides, because sometimes you need a couple of genes to make a couple of different polypeptides to make your mature protein. And then the other thing is, this actually is messenger RNA, but DNA codes for other types of RNA as well, like ribosomal RNA or transfer RNA. So the central dogma is that this is the chain of commands, but there are exceptions because DNA can code for other RNAs. And then there are retroviruses, which really mess this up. In a retrovirus, RNA makes DNA, which then makes RNA, which makes proteins. Okay, anyway, what you need to memorize on this is that, DNA to RNA is called transcription, and RNA to protein is called translation. Transcription, same language. If I told you to read TAC, you could go T goes with A goes with U, C goes with G, right? That's transcription. And then translation, you need your codon sheet for that. Translation's hard, it's a brand new language. AUG codes for methionine, AUC codes for isoleucine, UCG codes for serine, UAA is punctuation, it codes for a stop. So this is the beginning or maybe the end of some polypeptide, just as an example. Usually they're going to be a lot longer than that. But you need to know this general flow and know the transcription versus translation. The other thing to notice here is that 3 bases or 3 nucleotides or 3 letters code for 1 amino acid. So it's called a triplet code, where 3 nucleotides of RNA will code for 1 amino acid. So transcription uses the same language. So here's a doctor speaking into a recorder. Here's a medical transcriptionist that's listening to it and typing it out. So they are both in the same language. If I lose my job as a teacher, well, I'm not a great speller, but I do know English, pretty well. I could probably get a job in medical transcription, or probably not legal transcription, but maybe medical. I've got some terms. Same language, that's transcription. If I gave you T, T, C, A, G, T, you could tell me that if this is the strand that the RNA polymerase reads, T goes with A, C goes with G, A goes with U. I bet you could do this for me, right? Transcription, same language. So transcription is the synthesis of RNA from DNA, the language of nucleotides to the language of nucleotides, transcription. Translation is hard. 2 of the hardest subjects I ever took were Latin and Spanish. Some people are good at it, and I am not one of them. That's changing languages, though. I will never, ever have a job as a translator. It's just way too hard. So anyway, translation. You need a codon sheet for this. You could memorize it, I guess, but I don't have it memorized. AUG codes for methionine. I might have that, but I need to look at my codon sheet to go from AUC to isoleucine. So you will always have that codon sheet for you. All right. Next idea is the development of the gene expression idea. So the idea here is that everything we know in science is because some amazing scientists spent a lot of time trying to figure it out and doing experiments to prove it. Not just like, ooh, ho hum, I bet it's this way. They do experiments and figure it out. So early experiments showed that genes coded for enzymes. And so we knew from earlier experiments that DNA was a genetic material. But how does DNA end up making me? How does it give me blue eyes and one nose and two ears and all that stuff? And so the earlier experiments had, I think they were yeast, and they were given amino acids to grow on. And if you took out one of the amino acids, the yeast could still grow. But if one of them had a mutation, maybe it couldn't grow on that anymore. And so they said, oh, here's this mutation, this problem in DNA, and it ends up so that they can't. They don't have the enzyme that they need to make this other nutrient. So scientists said, oh, well, each gene must code for an enzyme. And so it's called the one gene, one enzyme hypothesis. And then later, experiments refined that a little bit to the one gene, one polypeptide hypothesis. All it means is that each segment of DNA codes for RNA, which codes for a polypeptide. That is generally true, although not 100%, because not all RNA is translated. Oops. Oh, I have this wrong. Not all RNA is translated into proteins. Whoa. Some RNA is regulatory, like for tRNA or ribosomal RNA, or like Pee-wee RNA or small interfering RNA, small nuclear RNA. So they are not all. That's not all. So the DNA to RNA to protein is not how it is all the time. Okay, so a gene is defined as a region of DNA that can be expressed to produce some final functional protein. This is kind of our working definition now. So we're not saying that a gene codes for a protein. We're saying a gene sometimes codes for a protein. So we're saying DNA codes for RNA, and sometimes it's messenger RNA and it codes for a polypeptide. So a gene can be defined as a region of DNA that can be expressed, used, right, shown, to produce a final functional protein. So that a final functional product. That product can be a polypeptide, or it could actually code for something else like transfer RNA. Transfer RNA is needed to make a polypeptide, but it doesn't give the code for a polypeptide. Or it could make ribosomal RNA, or small nuclear RNA, or Pee-wee RNA. We'll talk about all of these later. Or some other type of regulatory RNA. So this is actually our good working definition of a gene right now. It's a piece of DNA that codes for some product, which could either be a polypeptide or sometimes just a type of RNA.